Transmitters in particular, so this goes with 37.4, what happens in that synapse between neurons. And we'll also look at the nervous system, its responses. So how your nervous system allows response to different stimuli. So we'll start with the neurotransmitters and move on to nervous system responses. So what are neurotransmitters? Well, they answer the question, when an action potential reaches the synapse, what happens left? So here's the synaptic cleft, the space between the axon of one cell and the dendrite / cell body of the next neuron, right? How does that action potential get transported over this gap? So chemical messengers are released from one neuron and travel across this synaptic cleft, this synapse, and stimulate an action potential in the next neuron. So the neurotransmitters are those chemicals. They're released from the axon of one cell in response to a action potential, and then when they reach the other side, they create an action potential in the next. So neurotransmitters are stored in little vesicles right here, synaptic vesicles. And when the action potential reaches this area of the axon terminal, it stimulates calcium voltage-gated channels to open. And because these voltage-gated channels open, calcium rushes into this axon terminal. When the calcium is in this axon terminal, it binds to these synaptic vesicles containing the neurotransmitters. And because of that binding, these vesicles fuse with the presynaptic membrane and cause the neurotransmitters to be released into the synapse. The neurotransmitters then travel across the synapse and bind to ligand-gated ion channels in the next neuron. So here's a neurotransmitter that binds to this ligand-gated ion channel in the postsynaptic membrane, and its binding allows an influx of different ions, maybe sodium, maybe potassium, and this influx of ions creates the action potential in the next neuron. So then when the neurotransmitter is degraded or dissociates, it closes the synapse again, or it closes the ligand-gated ion channel in the postsynaptic membrane. So there are many, many different neurotransmitters that exist, tens and dozens, and they all have different jobs depending on the role of the neuron, if it's a sensory, efferent, afferent, in the brain, what part of the brain's circuitry system it's part of, and how it's stimulated, the frequency of stimulation. So there's different types and actions of some neurotransmitters. So neurotransmitters can be excitatory, so they can be abbreviated EPSP if they pass on this signal. So for example, we'll go back one. If they are released into the synaptic cleft and they cause this ion channel to take in ions that would depolarize the cell. So if it takes in sodium ions, then it is an excitatory neuron. It's causing a propagation of that signal. On the other hand, they might be inhibitory neurons or inhibitory neurotransmitters that cause the postsynaptic neuron to take in things like, go back here, potassium or chlorine, which hyperpolarizes that neuron and causes the signal to stop so that this next neuron doesn't propagate that signal. So, for example, neurons of the excitatory type open up sodium channels, and inhibitory open up potassium or chlorine ion channels. Some neurotransmitters don't activate an ion-gated channel at all, but are metabotropic. In this case, rather than an ion channel, it activates a secondary messenger in the receiving neuron, so it's more metabolic. A single neuron also might bind to many dozens of different receptors and be inhibitory or excitatory depending on the receptor that it binds to, and might actually elicit a different response, do something different in the body, depending on the receptor that it binds to. Right? Here's examples of how excitatory neurons might work. So, for example, if we're at the resting potential, and there are two excitatory stimulations in rapid succession, neither of them, if they're temporally distinct enough from each other, neither of them will excite past our threshold and create an action potential. So here we've got two different neurons trying to excite this neuron, but if those excitatory messages don't happen close enough together, it doesn't cross the action potential threshold and no action potential is propagated. If two responses from the same neuron happen close enough together, they actually can work synergistically and build on top of each other to elicit an action potential. Same thing with two stimulations from two different neurons that happen close enough together. However, if you have a signal from an excitatory and an inhibitory neuron, one can cancel out the other, and we can never make it past the action potential threshold. So some examples of neurotransmitters: acetylcholine, extremely important. It's a neurotransmitter that excites muscle cells. So it's your skeletal muscle cells, all your motor neurons contract because of their excitatory role, but acetylcholine can actually have an inhibitory role by reducing heart rate. So it has a different role depending on the channels that it binds with. Amino acids such as glutamate, glycine, GABA, which is abbreviation for gamma aminobutyric acid, are inhibitory, or IPSP, in most invertebrates. Biogenic amines, so dopamine, norepinephrine, serotonin, they have vital roles in regulating important aspects of both the central nervous system and the peripheral nervous system. And your endorphins, they're like natural painkillers that opioids like heroin try to mimic, and they have profound effects on pain perception in the central nervous system. Here in this table, I have some examples of neurotransmitters. So 1, 2, 3, 4, 5, 6, 7, and some of their roles. So not necessarily needing to memorize all of these, but if you see the name acetylcholine, recognize that it's a neurotransmitter. Acetylcholine actually is a good one to remember as far as its roles of being exciting motor neurons and relaxing involuntary smooth muscles, things like that. But this would be great to just look over, and it's nice to know, no need to memorize, but nice to be familiar with. All right. The nervous system is a giant circuit created by neurons and supporting cells. So in complex invertebrates, the nervous system is broken down between the central nervous system and the peripheral nervous system. Central nervous system, brain plus spinal cord. The peripheral nervous system, efferent and afferent sensory and motor neurons. So this is just kind of how we divide our systems of neurons into two functionally similar parts, right? But regardless, neurons aren't just hanging out there by themselves. They have a supporting cast, and one of them is called glial cells. And they are, in this diagram, kind of represented by brown and kind of these blue snowflakey like things, and what they do is they nourish and support the neurons and regulate their functions, and they're very important, microglia especially, in the development of neurons. So we can see here in our diagram we divide central nervous system and the peripheral nervous system. So we have the oligodendrocytes here creating the myelination for our central nervous system, the Schwann cells creating the myelination for our peripheral nervous system like we talked about last time. But here are those glial cells nourishing and supporting them. The PNS, peripheral nervous system, is composed of the motor system and the autonomic nervous system. So the motor system consists of neurons that signal skeletal muscles, so stimulate them to contract. And the autonomic nervous system signals involuntary responses and muscles, smooth muscles. So here is our central nervous system, and we have receptors that stimulate afferent neurons that send signals of us sensing internal or external stimuli to the central nervous system. Our central nervous system then takes that information, processes it, and sends out signals either to our motor neurons that control our skeletal muscles or our autonomic nervous system. It's broken down to our sympathetic nervous system, our fight and flight response, and our parasympathetic nervous system, our rest and digest response. And then the enteric division, which is basically has to do with our digestive system. But this is kind of a mapping of how we break up that circuitry between afferent neurons for stimulus input and afferent neurons in motor and autonomic nervous system functions. The brain is really important as far as the AP test concerns. The roles and functions of different portions, neuroanatomy, not very important. But the brain is important for integrating the information brought to it and then sending out orders of what to do. And the brain is divided into specialized parts that control specific functions. They're linked to the spinal cord and to the PNS via the spinal cord. So it's linked to the spinal cord and to the efferent and afferent neurons. The large portion of the cerebrum, so this part right here, cerebrum, is responsible for many of the higher functions and for many of the ways we interface with the world, like speech and smell and motor control. The cerebral cortex, the prefrontal cortex right here, for decision making, planning, logic. We have our cerebellum for coordination, fine motor coordination, things like that. The brainstem and midbrain, the reptilian brain, basic functioning, respiration, thermostat control, thalamus, our endocrine system, hypothalamus, pituitary, pons and medulla, aggression, things like that. So it's segmented and specialized to regulate certain things, to regulate or be responsible for regulating certain information processing pathways. Nervous system receptors transmit different internal and external data for processing. So sensory receptors transmit sensory information from the body to the CNS. So we're talking about this section, sensory receptors. So how we sense information, internal and external. Many of these receptors are sensory neurons, so actually neural cells, but some are cells that just regulate sensory neurons. So here is an actual sensory receptor that via stimulation induces a action potential to the CNS. In a sensory receptor cell, stimulus leads to neurotransmitters being released from a non-neuron, and that then stimulates a neuron that has a receptor, an afferent neuron, that then continues the pathway of the CNS. Changing presence or absence of a stimulus to an action potential is called sensory transduction. So this, in both parts, is sensory transduction right there, changing stimulus to an electrochemical signal. The greater the stimulus, the more frequent the action potential it generates. So for example, if we have gentle pressure, we'll stimulate a action potential less frequently. If we have greater pressure, it'll be a higher frequency in that action potential. Now, on its trip to the brain, to the central nervous system, amplification happens, which is increasing the strength from this initial signal to a very strong signal. That's why your eyes can actually respond to a single photon of light, because this amplification of maybe a very weak signal from one sensory receptor can be recognized by your whole central nervous system. Of course, different sensory receptors, like those in your hands versus those in your eyes, those on your tongue or your nose or in your ears, are able to connect to different areas of your brain. So this is why we perceive touch and sight and smell and taste and hearing differently. They eventually stimulate different areas of the brain that interpret those signals differently. So, some examples. We have many different types of sensory receptors. Mechanoreceptors, which perceive touch. Pain receptors, also called nociceptors, perceive pain. Thermoreceptors, that's heat or temperature. Chemoreceptors, chemical substances, so smell, taste, things like that. Gravity sensation, mechanoreceptors present in statocysts, which are actually in invertebrates. Hearing in the inner ear and equilibrium via several different cell parts or anatomical features of the ear, the urticle and the saccule. So, some examples here in the ear: when sound waves cause the fluctuation of the tympanic membrane, it creates a mechanical vibration that goes from the malleus and incus to the stapes into the semiocular canals in the cochlea. And there's hairs in the cochlea that vibrate, and these vibrations of the hairs in the cochlea begin to open ion channels and stimulate an action potential in the auditory nerve that leads to the brain for perception of sound. Same thing with why we can have mechanoreceptors in hair. The bending of hairs in one direction can release neurotransmitters into a receptor neuron under the skin, and that's how we feel the breeze through our hairs. Bending of the hairs in another direction can cause less neurotransmitters to be released, and then we see a space or a decrease in frequency. So a less strong breeze, a more strong breeze. That's how mechanoreceptors, your hair, can give you the sensation that there's a breeze or there's a wind flowing through it. They lead to the release of neurotransmitters in these portions right here, the living cells, to sensory receptor neurons or nerve cells. And last, I want to talk about vision in the eye. So in your retina, the disc membrane inside retina, you have a chemical called rhodopsin. Rhodopsin in the cone cells, so this is rod cells. So this is a rod cell in the eye. Rhodopsin is a chemical that is activated by light. So when light hits rhodopsin, it activates rhodopsin. And active rhodopsin signals a transducin protein to activate phosphodiesterase. What phosphodiesterase does is it inhibits adenyl cyclase from turning guanosine monophosphate into cyclic, or guanosine triphosphate into cyclic guanosine monophosphate, or CGMP. What that does is it closes a sodium ion channel. Which then hyperpolarizes the cell and leads to the absence of an action potential, leading to a message being sent through your optic nerve to your brain, to the visual cortex in the back. So we've got, let's see, we've got, pretend this here. There's our optic nerve. So this secondary messenger pathway these chemical messengers can actually cause you to see. And this is an example of a metabotropic action of sensory reception, right? So this secondary messenger pathway can cause an action potential to be created in your optic nerve, which then can cause your brain and your visual cortex in the back and your occipital lobe to perceive a sight, right? So hopefully these several examples help you to figure out the different ways that your brain and your nervous system respond to stimuli or integrate stimuli in your central nervous system. And we'll talk about this more on Wednesday.